Hello everybody. Today we shall continue the chapter with pollen pistil interaction. As you know that we are studying the sexual reproduction in flowers, and in that we have studied the flower, the development of male gametophyte, development of female gametophyte, then pollen grain, pollination, and now we are going towards the study of the process of fertilization. And in this series of the sexual reproduction study in angiosperms, today we shall study pollen pistil interaction. What is pollination? The transfer of pollen grains from the anthers on the receptive stigma is called as pollination. The pollen grains are deposited on the stigma at two-celled stage. One is the tube cell, the larger cell, and the smaller is the generative cell. And in most of the angiosperms, at the two-celled stage, the pollen grains come on the receptive stigma. This process of transfer of pollen grain is pollination. Now what happens when the pollen grains comes on the carpel? The stigma, style and ovary are the parts of a typical carpel. What happens to these pollen grains? It is to be studied in pollen pistil interaction. Pollen refers to pollen grain, pistil refers to ovary style stigma, and their interaction is to be studied here. The process of pollination, where the pollen grains come on the stigma, is a chance method. Any pollen grain present in the air or stick to the insect may come in contact with the stigma. Maybe of the same species, maybe of the other species. The pollen grain may be of the another genera, may come on the receptive stigma of a plant. But the interaction between the stigmatic chemical substances and the pollen grain deposited on the stigma takes place only in between the intraspecific pollen grains. The pollen grain here may be of the same species or another species. If the pollen grains deposited on the stigma are of the same species, then we call it as a right type of pollen grain or a compatible pollen grain. And if the pollen grain is of other species, other genera, may happen. Pollination is a chance method. Then the other pollen grain is a wrong type of pollen grain, incompatible pollen grain. The wrong type, the incompatible pollen grain, cannot develop further on this stigma. The right type can be, or the compatible type of pollen grain, which is intraspecific of the same species, can further germinate on the stigma. The compatible pollen grain absorbs necessary chemical substances from the stigma as well as water through the germ pores which are present in the exine. And when it absorbs water and necessary chemical substances, pressure is created in the pollen grain from the inner side. And now, due to inner pressure, the intine, the inner layer of the pollen grain is called intine. The intine from the germ pore, it comes out in the form of a tube. Germ pores are present on the exine. Inner layer is intine. The intine comes out through the germ pore in the form of a tube called as pollen tube. Now this pollen tube initially grows or develops on the stigma. Then the path of the pollen tube is followed into the hollow style, and from the style, the movement of the pollen tube takes place towards the ovary and the ovules in the ovary. Many pollen tubes can develop on the stigma, and many may enter into the ovary because many ovules may be present within the ovary. Now such pollen tubes, while germinating or developing on the style and the stigma, the generative cell which is present in the pollen tube, it divides by mitosis to form two male gametes. Now this structure, the pollen tube, the two male gametes, the pollen tube along with the tube nucleus and the two male gametes, pollen tube along with the tube nucleus and the two male gametes, collectively the three-celled structure is called male gametophyte. Now this development of male gametophyte mostly happens only during the process of pollen-pistil interaction. The tip of the pollen tube is followed with a tube nucleus. It is followed with two non-motile male gametes. This content is taken towards the ovule by the developing pollen tube, and thus the pollen tube then reaches the ovule and the further process of fertilization is carried out. Until the process of the pollen tube reaching to the ovule is studied as pollen-pistil interaction. What is studied in pollen-pistil interaction? Transfer of pollen grain from anther to the stigma is called pollination. This takes place at two-cell stage of pollen grain. Only the compatible, the right type of pollen grain, intraspecific type, can further germinate on the stigma. The wrong type pollen grain, which is not compatible, cannot germinate. Only compatible can germinate. Incompatible cannot germinate. Wrong type is discarded. The pollen grain absorbs water, necessary chemicals on the stigma. Intine comes out in the form of a tube, grows on the stigma, develops in this style. Meanwhile, two male gametes are formed from generative cell by the process of mitosis, and the gametes are carried into the ovary towards the ovule. This all is studied in pollen-pistil interaction. With the development of the pollen tube up to the ovule, the next step is of double fertilization. What is double fertilization? Why do we don't say only fertilization here? Double fertilization, the word itself means that this is the characteristic feature of angiosperms. Always in angiosperms, the fertilization is double fertilization. What does this double fertilization means? Of the two male gametes in the pollen tube, one male gamete unites with a haploid egg to form a diploid zygote. Another male gamete unites with a secondary nucleus to form a triploid primary endosperm nucleus. As two male gametes take part in fertilization at two different places, it is called double fertilization. What is double fertilization? Of the two male gametes, one unites with, remember that all the gametes are haploid. The male gamete, haploid N, female gamete N. If it is diploid, we say 2N. The gametes are haploid, therefore N. One male gamete unites with another female gamete to form a diploid zygote. What is formed? Diploid zygote is formed. Another male gamete unites with secondary nucleus. The secondary nucleus is diploid. Why do we say the secondary nucleus is diploid? Because before the formation of secondary nucleus, there are two haploid nuclei, polar nuclei. This is secondary nucleus. Before this secondary, there are two polar nuclei. Polar nuclei are N plus N. The N plus N, when they unite or fuse, now the polar nuclei after fusion are called secondary nucleus. The secondary nucleus is the only structure in the embryo sac or in the female gametophyte which is diploid. This secondary nucleus is diploid. All the other structures are haploid. So this 2N of secondary nucleus, then male gamete N, it forms 3N, which we also called as triple fusion or primary endosperm nucleus. So, while studying double fertilization, first of all we have to understand that double fertilization is the characteristic feature which happens in all the angiosperms. All angiosperms. There is no single fertilization, it is always double fertilization. What does this double fertilization mean? How does it take place? Of the two male gametes, in the pollen tube, one male gamete unites with the haploid egg. Haploid male gamete unites with the haploid egg to form diploid zygote. The second male gamete unites with the secondary nucleus to form a triploid primary endosperm nucleus, hence called double fertilization. We can also say that as two male gametes take part in fertilization at two different places within the embryo sac, the process is called double fertilization. The first to study in double fertilization is the definition of double fertilization. Second, the word syngamy is important. What is syngamy? The fusion of first male gamete along with the female gamete is known as, to form the diploid zygote, is known as syngamy. The fertilization of the second male gamete with the secondary nucleus to form triploid primary endosperm nucleus is known as triple fusion. These two form double fertilization. So now, how the process is carried out? When the pollen tube containing the two male gametes enters in the ovary, it enters into the ovule. How does it enters into the ovule? Most of the times, the pollen tube enters the ovule through the micropyle. This pollen tube, it enters into the ovule through the micropyle. It is called as porogamy. When it enters through the micropyle, it is called as porogamy. In some plants, the pollen tube may enter into the embryo sac through the integuments. When the pollen tube enters into the embryo sac by piercing, by entering into the embryo sac, then the process is known as mesogamy. Sometimes, in some plants, the pollen tube, it enters through the chalaza up to the embryo sac. When it enters through the chalaza up to the embryo sac, the process is known as chalazogamy. So, the entry of pollen tube into the ovule is of how many methods? 3 different methods. If the pollen tube enters the ovule through the micropylar pore, it is called porogamy. If the pollen tube pierces the integuments while entering into the embryo sac, then it is called as mesogamy. If the pollen tube enters into the ovule through the chalazal side, it is called as chalazogamy. Now, whatever may be the pathway, either may be the porogamy, mesogamy, or chalazogamy, the entry of the pollen tube in the embryo sac, the entry of the pollen tube in the embryo sac is through the filiform apparatus. It is through the filiform apparatus which is present, filiform apparatus which is present at the tip of the apex of the egg apparatus here. The pollen tube may come through the integuments, may come through the chalaza or the micropyle, but its entry in the embryo sac is always through the filiform apparatus which is present at the tip of the egg apparatus in the embryo sac. This filiform apparatus has some chemical stimulus which attracts the pollen tube towards it. And from the filiform apparatus, the pollen tube now enters into one of the synergids. When it enters in one of the synergids, this pollen tube absorbs water and necessary chemical substances here. Till then, the tube nucleus which is present at the tip, it degenerates, it disappears. Now, at this stage, only two male gametes are present in the pollen tube. Tube nucleus at the apex degenerates. The pollen tube now contains only two male gametes. The pollen tube, when it absorbs necessary chemical substances and water in one of these synergids, it bursts open to release the male gametes. Pollen tube breaks and bursts and throws out the two male gametes in the embryo sac in between the cells of the female gametophyte. When the male gametes come out, one male gamete, the first one fuses with the egg to form diploid zygote, and the second one fuses with the secondary nucleus to form triploid primary endosperm nucleus. The technical term for the formation of the first fertilization, the diploid zygote, the future embryo, the technical term for this fusion is called syngamy, and the technical word for the fusion of 2n plus n, secondary nucleus plus n of male gametophyte, is triple fusion. This is how the process of fertilization is carried out in the angiosperms. Now here another point we have to note is both the gametes, male gametes or maybe the female gamete, all of them are non-motile. Now as the non-motile male gametes are brought up to the female gamete with the help of a tube. With the help of a tube, they are brought up to the female gamete. Hence, the word is known as siphonogamy. Hence, called as siphonogamy. What is siphonogamy? The pollen tube is called siphon. Tube refers to siphon. As the non-motile male gametes are brought up to the female gamete with the help of a siphon, a tube, the process is described as siphonogamy. Now with this act of fertilization, diploid zygote is formed in the region of egg, and primary endosperm nucleus is formed with the act of triple fusion. The further changes with the formation of zygote and the primary endosperm nucleus are studied in post-fertilization changes. So, up to the formation of syngamy, the zygote, and the triple fusion leading to the formation of primary endosperm nucleus can be called as PEN, P capital, E capital, N capital, primary endosperm nucleus. Until this zygote and primary endosperm nucleus, it is double fertilization. The next step in the study is post-fertilization changes. After fertilization, post-fertilization changes. Who was the scientist who first discovered the process? The process of double fertilization was first of all discovered by a scientist called Navastin. And Navastin discovered this first in the family Liliaceae, mostly in the plants such as the Lilium and Fritillaria. Scientist Navastin was the first person to discover the process of double fertilization in Liliaceae plants monocot family, and the plants where he studied this was Lilium and fritillaria. What is the significance of double fertilization? It is the first significance is that it is the characteristic feature of only and only angiosperms, flowering plants, angiosperms, always double fertilization. So first line is insignificance, it is the characteristic feature of double fertilization. What is the second significance? The zygote, the first fertilization, zygote leads to the development of embryo of the seed. Then, what is the significance of triple fusion here? The primary endosperm nucleus, result of triple fusion, it develops into endosperm of the seed. So, with the formation of embryo from the zygote, endosperm from triple fusion, the entire ovule, this ovule slowly changes into seeds. Seed. Single ovule, single seed. Many ovules fertilized, many seeds develop into the ovary. And when seeds begin to develop in the ovary wall gets converted into fruit wall, and finally, after fertilization, there is formation of seed from the ovules and fruit from the ovary. So what is the significance? What are the significances of double fertilization? It is the characteristic feature of angiosperms. Syngamy, the first fertilization leads to the formation of zygote, which develops into embryo. The triple fusion leads to the development of primary endosperm nucleus, which later develops into endosperm. In the process, the entire ovule gets converted into seeds and the ovary gets converted into fruit. This is a general significance during the study of double fertilization. In the process of double fertilization, what we have to remember here is the first thing is definition. Two male gametes fertilized at two different places, one with egg, another with secondary nucleus, hence called double fertilization. Then the word syngamy, then the definition of triple fusion, then what is porogamy, what is mesogamy, what is chalazogamy, what is siphonogamy. When we write sentences for all these 7 different words, we can describe the entire process of double fertilization in angiosperms. And depending on these words, we can also explain the significance of double fertilization. So today, we have studied the pollen-pistil interaction first. The pollen-pistil interaction later helps in process of double fertilization. Then the double fertilization later slowly and steadily converts the ovule into seed and the ovary into fruit. But all the changes in zygote and the triple fusion are studied as post-fertilization changes. So in the next class, we are going to study post-fertilization changes in the ovule and the ovary, as well as the development of endosperm embryo also within the ovule. Thank you.